do.
Okay, so I'll start. So next presentation is on insect vectors in Yucatan and collaborators from CC. And for background, jump this and this. And well, this is a very nice diagram showing how it is supposed that the phytoplasmas can be vectored by insects into plants. And well, what has been reported before and within the Tropicsafe project, but not within this presentation. Well, in the USA, it was reported that phytoplasmas were present in Aplaxus crudus, but it was not done molecularly. And then in Jamaica, they reported Sedusa, Cuba, Nymphorixia, and Mexico, Aplaxus crudus. And in all cases, the phytoplasmas were reported as group 4, but without subgroup. In the case of Mexico, it was already being reported subgroups A and D. Then within the Tropicsafe project, there are reports on the Plaxius, Hortensia, subgroup A, and Aplaxus crudus, Capion, Catueli, Snowy, and Persis. All three of them with A, and the other two are still to be defined the subgroup. Pictures of some of them are here. In regards to transmission, the first report was that from Howard and colleagues, several reports rather, showing that Aplaxus crudus, known by that time as Indus crudus, was able to transmit phytoplasmas to different palm species. And this was carried out using insect-proof cages and detection by electron microscopy. Then in Yucatan, there were attempts to transmit phytoplasmas using Aplaxus crudus, and it was successful with Pichardia pacifica palms, and the subgroups were A and D. And in the case of Aplaxus, same, A and D to Cocos nucifera. In Tabasco, Carlos's Freddy group reported the transmission of phytoplasmas by Aplaxus crudus to Adonidia merili, and the subgroup was A. Some pictures of the experimental work. And the question is, are white phytoplasmas transmitted by insects other than Aplaxus crudus, or it is only Aplaxus crudus? Well, results. Well, again, I have already talked about this. Just would like to mention that to stress that the fact that in this plot, the amount of plants dying was high. In the case of the segregating population, it was 46%, and in the case of the Atlantic toad, it was 93%. But in regards to the current subject, it was interesting that in that site during the outbreak, what was found in looking at what insects were present. You can see here the total number of insects, but then basically with the same pattern, the Colpoptera insects, but very interestingly, the percentage of Colpoptera within the whole population was about 1/2, so very high. And other thing is also relative to what we are trying to do, finding whether there are other vectors. This insect was positive, so that could make us think that it could be a vector. But the other thing that was very important is that in this outbreak, no screws were found. So somebody else should be guilty. Okay. So in regards to detection of Colpoptera, it was 55%, 25%, sorry, of insects that were positive, which is a high percentage if we compare with what was reported previously for Aplaxus crudus in Yucatan, less than 2%. And the phytoplasma found in Colpoptera was group 4, subgroup A. We also found another insect species that was positive, no subgroup defined yet. Percentage high, but it was a very small sample. So this is yet to be studied further. The interesting case now is Colpoptera because it was very abundant, because it was positive in large percentages, and because no black screws was present. So it's a candidate. Then a first trial was carried out using the traditional methods as Howard's in insect-proof cages, only that in order to try to reduce the number of insects tested, we use a device so we could try the transmission with small numbers. The results from this test were negative, but what we found is that when analyzing the insects, they were negative. So unfortunately, during this period, the insects collected were negative. And well, that was before the pandemic. Then after the pandemic, we did another test. During the pandemic, we couldn't do anything because we were not allowed to go to do field work. Collecting insects is something that has to be done every week. In this second test, what we did was to use a different kind of transmission system, which is in vitro. This was successful previously for Aplaxus crudus, so we tried it. And what we did was to use 6 plants for Colpoptera, 6 plants for Aplaxus crudus as a reference, and 6 plants for no insects introduced. The number of plants was small because it was a test carried out at the end of last year, and we are planning to do it again at least two more times. Well, in this case, we found that there was a positive case of detection of phytoplasmas in the plants, but also in the insect. And in the case of Aplaxus, it was the same. It was 1 of 6, but we had positive results. So concluding this study, about 10 species have been found infected with 16SR4 phytoplasmas in different countries in the Americas, including 6 of these in Tabasco and Yucatan in Mexico. 2 species found in Yucatan, Homoligna and Colpoptera. This is besides Aplaxus, and were discovered as part of the Tropicsafe project. Colpoptera was considered as a candidate as a vector of these phytoplasmas because it was associated with the LY outbreak in north of Yucatan. It was very abundant, and a large percentage of them were infected. Preliminary results show that the Colpoptera is associated with phytoplasmas, group A, and that it was capable to transmit them to at least 1 coconut plant. Further research is required to fully confirm that Colpoptera insects are capable to transmit these phytoplasmas and define their relevance in the epidemiology of LY disease. Thanks very much.